No, I am just recording something here.
I've seen. Wait, are you like recording it right now?
Yeah, but it's not recording my voice. It's just a screen recording.
I've seen so many memes about the Capitals' defense was like worse than the Cowboys.
Oh God.
It's kind of funny, but it's kind of wrong.
It is. It is.
You see, well, the special thing is the stupid name of Chargaff rules. I've never quoted Chargaff rules, but anyway, that is the base pairing. I quoted complementary situation. I don't think I gave the name base pairing. I think that is one of the few things that Watson really did.
The DNA molecule is made up of very long chains of the four bases A, C, G, and T. In 1950, Erwin Chargaff published a paper stating that in DNA of any given species, the ratio of adenine to thymine is equal, as is the ratio of cytosine to guanine. This is known as Chargaff's ratios, and it was a crucial clue that helped solve the structure of DNA. Chargaff's ratios are universal. All forms of life obey this rule. Only the balance of AT pairs and CG pairs varies between species. This is Linus Pauling's failed attempt to predict the structure of DNA. The problem with his triple helix model is that the phosphates form the helical core, with the bases pointing outwards. This would be impossible under normal cellular conditions. Each phosphate group is negatively charged, and so many negative charges forced together would repel each other, literally driving the structure apart.
That evening, sort of 6 o'clock, we went over when the Eagle opened and, had a drink, more or less, to toast Linus's failure. It was, because, we never contemplated. We thought, well, propose a model, and it might not be right. But never did we think, well, one that we could reject so instantly. And so we were both pleased and a bit scared because we thought it was, just awful. Maybe someone at Caltech, upon reading the manuscript, will tell Linus this is chemical nonsense. Little did we know that, no one at Caltech really had the courage to tell Linus he was wrong. No matter how good a school it was, Linus was like the Pope, and it wasn't a way to, Linus wasn't used to people saying he was wrong.
Wait, what? William, what?
Rosalind was tickled pink because we'd been working away and we knew from the way we could take water in and out of the structure and further change the structure from one crystalline to a paracrystalline thing with ease, that the things that the water were attaching to was the sodium and phosphorus. You had the phosphate group and the sodium sitting there, and that attracted a whole lot of water molecules around it. So the water going in and out meant that the phosphoruses must be on the outside. And so she laughed at them, much to their discomfiture, I think, and said, "Oh, look, you've got it inside out. The phosphoruses are all on the outside." And they are for the following reasons. And she explained her experimental work and her reasoning, and so they sat back and absorbed that and suitably chastened. They, I don't know what they did. They probably didn't do anything for a while. And they never got together from the word go to work as a team. Partially, I think, because they were constitutionally more suited to working separately. I mean, Uncle Morris was, as we called him, not necessarily to his face, but other PhD students in there, we all referred to him in this way as being an elder statesman who was above us all and very clever, and you will go along. But we weren't working hand in glove. There wasn't that intimacy, if you will, that developed between Watson and Crick. Wilkins was shy and kept himself. Along comes Rosalind Franklin, thinking she is going to do a particular job, also being rather reserved and wanting to get on with it. And they just did not mix from the word go. They never sat down together long enough to exchange ideas.
the cabinet shop was to build us some tin models, and that took too long. And, finally in desperation, I made some out of cardboard. I began moving them around. And I wanted an arrangement, where I had a big and a small molecule. And so how did you do it? Somehow you had to form link bonds. So here's A, and here's T, and I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Whew. So then I went to the pair. I wanted this nitrogen to point to this one. I went like this. Whoa, they look the same. And you can put one right on top of the other. I guess my chief advantage came from coming from the field of genetics, where the big question is, what is the gene? And I sort of boxed myself in, and that was the only problem worth solving. Whereas Rosalind was trained as a physical chemist, Morris as a physicist. They didn't see themselves, as having only one objective. So my advantage, one was, it was the only thing I wanted to do. And second, the other advantage was I had a collaborator with Francis. So one can't underestimate the importance. If I'd come to Cambridge and Francis wasn't there, I wouldn't have found the structure of DNA. I wouldn't have, had someone to talk about the meaning of the X-ray diffraction. And Proust and Kendall were interested in proteins. So Francis saw DNA as tremendously, I should say, Francis saw DNA as potentially, the most exciting molecule around. When the Nobel Prize was awarded in '62, they waited to '62, I think, really because a lot of the people thought, well, it's a pretty model, but maybe it isn't really the model for how DNA replicates. With the Meselson-Stahl experiment, then the model was right, and 4 years later we got the Nobel Prize. But then Rosalind was not living, and so they had the rule only 3 people can get a given prize. So there wasn't any problem. Morris got one, I got one, and Francis. If Rosalind had been alive, the only sort of just thing would have been 2 Nobel Prizes, one for Wilkins and Franklin for the experimental work, and one to Francis and I in biology. They could have got the prize in chemistry, and we could have got the prize in physiology or medicine. That would have been, everyone would have been pleased.
I read that pair of papers in Nature. I was impressed. It didn't alter my way of life an iota. And in fact, there is a rather naive component in that paper, which assumed that once one put the individual A next to a T, and successively each of the building blocks, somehow they link together spontaneously. And to anyone who'd worked with enzymes, this was so naive and impossible, because you depend in every chemical reaction in nature, even the hydration, adding water to carbon dioxide, on an enzyme to catalyze that reaction thousands of fold faster than it ordinarily would, and to direct it. And so we already knew that enzymes had to exist to catalyze and direct the assembly of a double helix.
Well, we're trying to find out if DNA replicates semi-conservatively, or dispersively, or conservatively. And that means, do the two strands separate but remain as single strands indefinitely, and each become associated with a newly synthesized strand at each generation? Or do they come apart and get dispersed? Or do they stay together, the two strands, indefinitely, making only brand new DNA molecules alongside themselves?
Yeah. Just recording something here real quick.
We start the experiment by growing the bacteria for a long time, many hours, many generations, in heavy nitrogen medium. So everything's heavy nitrogen. And now we put a little bit of that bacteria into some dupanol, some detergent to liberate the DNA. We put that in the ultracentrifuge, and we centrifuge it, and all the DNA is heavy. It forms a single band. Meanwhile, most of the bacteria have been left behind in the culture tube, and we centrifuge them down and resuspend them in light nitrogen medium. So now any more DNA that's going to be made is light, not heavy. And we see that as time goes on, there builds up a new band until at one full generation after the transfer from heavy to light, all of the DNA is at this new density, which is the density we expect for DNA with one heavy strand and one light strand, namely hybrid DNA. And then as time continues to go on, back in the culture tube where the bacteria are growing in light nitrogen medium, still another band begins to appear. That's fully light DNA. Both strands are nitrogen 14, the light kind of nitrogen. And now as time goes on, the heavy strands that are inherited from way back there are never destroyed. They're, in a sense, immortal. But as more and more DNA is made, it can be made only out of light DNA. And so the light DNA, two strands, both light, begins to become more and more predominant in each sample until we ran this experiment, in this case, to 4 generations.